now that you know about DNA and proteins and how they all work together to give us traits and organisms, we are now going to talk about some of the practical applications of that, okay? And so we're going to be learning about genetically modified organisms today. And you might have heard that word, they're called GMOs, okay? And you may or may not really have a good handle on what they are, and that's okay because we're going to figure it out today. When a lot of people hear the word GMO, they're thinking it's something like this. Ah, right. Like Frankenfish. Ooh, part apple, part goldfish. No, this isn't a thing. It doesn't exist, right? Like Photoshop. Um, but this is not what people are talking about, okay? You can't make a plant animal hybrid. It wouldn't live. It's not viable, okay? Um, and so in recent years, we have developed the technologies to modify organisms, okay? But actually, even before technology, we have been modifying or slightly changing the genetics of organisms without like fancy pants, like microscopes and, test tubes and lab coats and goggles and all this stuff. We've been doing this for tens of thousands of years, okay? The simplest way that you can modify an organism is through selective breeding, okay? And this is just when humans take a look at the things around them and they pick the traits that they like and they make more of those things, okay? So for example, cows. We rely on cows for dairy. Well, if you've got a cow that's a really big dairy producer, guess what? You're going to mate that cow and have that cow produce a whole bunch of babies. And so then those babies should hopefully then, right, that cow's going to pass on that high producing milk trait to its babies. And now the next generation of cows is going to produce more milk. And then you take those highest producers and you mate them and so on until you get super high producing milk, right, over generations and generations of cows. Okay. So humans have done this a lot because we want that certain characteristic. In this case, it's milk production. Okay. And this is a form of genetic modification because we are making sure that certain genes are showing up and becoming more and more popular in a given species or a population, right? By which ones we choose to breed, which ones we don't. And we might take two real high producing cows, like a cow and a bull, and put those together, right? To produce super crazy high producing ones, right? So we are artificially doing that. We're not letting the cows pick their mate. We are picking the mate for the cows to give us this desired trait. Some other examples of where humans have done this would be corn. Okay, ancient corn, the ears of corn. So here's a quarter. Whoa, so the ear of corn is roughly 2/4 big, and it produced these teeny tiny kernels that were like barely edible. Okay, but every once in a while, one of these plants would produce some kernels of corn that were like a little bit bigger, and a little bit like juicier. And so you would take that plant and breed it with another plant that kind of produced some bigger ears, and so on and so on and so on. Always producing the ones, always breeding the ones that had like the bigger ears with the less like crunchy stone-like corn, and keep breeding the good ones over and over again until we now have our modern day corn where here's the size of a quarter compared to a modern day ear of corn, right? So we as humans have changed the size of ears of corn, and also the kernels are now nice and plump and full of water, whereas it used to be like really, really, really dry and crunchy. Okay. So we've done that by picking which ones to breed. Same thing with dogs. Okay, think about all the different dog breeds. Humans brought those to like exist, okay? Back in the day there were wolves, okay? And humans, this is a little Game of Thronesy, but some wolves started hanging around some pack of humans, and if you got like a really nice smaller wolf and another really nice smaller wolf, well, maybe you'd breed them together to try to produce some other nice small wolves that aren't going to like rip your face off, okay? And so in different areas of the world, they would produce the nicer wolves and they would breed them together. And then, certain people said, ooh, I want my dog to help me hunt. And so, ooh, in this litter of puppies, this was a good hunter. So I'm going to mate that one with another good hunter, and I'm going to make a hunting dog. Or, ooh, I want a little teeny tiny dog that's going to sit on my lap. So I'm going to take this teeny tiny dog and this teeny tiny dog, and I'm going to put them together so they have even teeny tinier babies, right? So this is how we get all of the different dog breeds that are seen today, like a Labradoodle. That was humans. It didn't exist until recently. Dalmatians have been bred for their spots. Okay. All of the different dog breeds that we see are because humans have just taken dogs with characteristics that they liked and bred them together over and over and over again. Okay. However, when people talk about genetically modified organisms, they're typically not referring to this, right? Like this feels like, okay, well, we're just taking two things, we're mating them, but like, we're not like changing them that much. Okay. So what people usually get fired up about is not selective breeding. Okay. What people sometimes get a little bit fired up about, or they get a little bit like, I don't know how to feel about this. That's when we get into genetically modified organisms that are produced through genetic engineering, which is what we're going to talk about next. Okay. So basically with genetic engineering, what you're doing is you are taking a gene that a thing doesn't have and you're adding it to that thing to give it some type of trait you want. Okay, so this is not taking two nice wolves to have like really nice wolf babies. Okay, this would be like, hey, have you ever heard of diabetes? Okay, diabetes comes about because some people can't produce insulin properly. So guess what? We figured out a way to have bacteria produce insulin, and then we harvest it and we give that insulin to people with diabetes. Cool. Okay, so we gave the bacteria the insulin gene, and so now it can make insulin for us. Okay. We have taken rice and we have added a gene into it with a vitamin that helps reduce blindness. Okay, especially in developing third world countries where the nutrition isn't so great. So again, this rice didn't have the vitamin before. We took this vitamin gene, popped it into the rice, and now it has it. So anyone who eats the rice now gets this vitamin. We've also engineered a lot of our crops so that they can be resistant to a herbicide. Okay, herbicide kills things like weeds, but they could also kill our crops too. So we want the herbicide to kill the weeds, but not the crops. So if we put this herbicide-resistant gene in there, now all the weeds will go away, right? We'll have less of our crops die, and we'll get a higher yield from our fields. Okay, so these are all ways we could take a gene that a thing didn't have before and pop it in there to give us a new trait. So let's zoom in now and see the steps involved and how do you actually do this. Step number 1, you want to find the gene of interest in the DNA of some organism, okay? And then you're going to find that gene. So maybe it's the insulin gene, okay? So I'm going to find that insulin gene in humans, and I'm going to use this thing called a restriction enzyme. Like, let's say here's a gene right here, obviously simplified. I'm going to boop, boop, snip it out with these things called restriction enzymes. And I'm going to cut this gene away from the rest of the DNA. Okay, how do I do that? Well, so this restriction enzyme, basically an enzyme is just like a protein helper that helps something happen, like a reaction happen. Okay, and these restriction enzymes are like teeny tiny DNA scissors. And they just snip DNA, but they don't snip DNA in any old place. They snip DNA at very specific parts of a DNA sequence. So let's say right here, this is the gene that we want. That's that insulin gene. There's lots of different enzymes. One's called EcoR1. Okay, EcoR1, when it sees this specific GAATTC, it's going to snip there. GAATTC, it's going to snip there. So notice if our enzyme snips, ooh, look, we've got our gene that we wanted right there in the middle. Yeah, so that's like the zoomed in version of this. So that's why it snips there, because this EcoR1 enzyme snips whenever it sees this sequence of DNA. Okay, more on this later. You'll be doing more with this later. Okay, so now we found our gene. We use an enzyme to cut the gene out, and now we're going to put the gene into a plasmid. Okay, what's a plasmid? Well, if you remember back to the beginning of the semester, we learned about prokaryotes and eukaryotes and how prokaryotes typically have one circular chromosome, like right here. Okay, prokaryotes oftentimes have little bitty bits of DNA called plasmids, okay? And they're just little circular rings. They're in bacteria, fungi, and actually in some plants too, but typically in prokaryotes. And they're free-floating in the cytoplasm and completely independent from the rest of it, okay? And so what we're going to do is we're going to take this circular piece of DNA. I'm moving up here. I'm going to take my circular plasmid. I'm going to pop it open with another enzyme that's going to snip. Ooh, I'm going to take that insulin gene and I'm going to pop it in and use another enzyme to glue the piece in place. And so now ooh, my circular plasmid now has that gene I want in it. Cool. Very cool. Next, okay, I got my gene in my plasmid. Now I got to get my plasmid into the thing, right? Whatever I'm trying to change. And so this is how you could do it with a bacteria. Here you've got your plasmid with maybe your gene of choice. Here's your bacteria. And you can expose it to like extreme temperatures and chemicals, or you could pass like an electrical current through it or something, and that's going to cause this bacteria to bloop, bring the plasmid inside. Ooh, and now it not only has its normal DNA, but it has this extra DNA with that gene that we want. Very cool. Okay. And then lastly, now we're going to let that organism grow and multiply. And hopefully, if we've done it right, now that thing is going to have the trait. Now that bacteria is going to be able to produce the insulin, let's say. Okay. So here's our bacteria. Here's the plasmid with the insulin gene. And so now we should start to see that bacteria and all of its babies be able to produce insulin themselves. Okay. And you all are actually going to get a chance to do this in lab. You'll be doing this. So more on this in a bit. Here is a real life example of how we've done this with corn. Okay, so we want our corn to be resistant to this certain type of caterpillar so the caterpillar can't eat it. Okay, so what we did is we found a gene in a special bacteria in soil. Okay, we found that gene, we snipped it out with a restriction enzyme. Okay, we put it into a plasmid. We let that thing grow and multiply, okay? And then actually a neat thing is, well, how do we get that into the corn then? One of the things that they did is they literally like blasted it with gas under high pressure, like, and like bombarded the corn with this bacteria that had our gene in it. And then our corn then got that DNA and incorporated that DNA in it. And so now our corn has that trait of being resistant to this caterpillar from eating it, which is kind of cool. Now, you don't need to like memorize this or anything, but this is just an example, simplified version, of course, of how we have actually done this in modern day. So, genetically modified organisms, what do we think? Well, you've probably almost certainly eaten some in your lifetime. Okay, so 94% of the soybeans produced in the United States are genetically modified in some way, somehow. Again, maybe they're just resistant to some kind of pesticide or some kind of insect, but that's modification. 94% of the cotton grown in the United States, GMO. 92% of the corn is GMO, and that corn goes to feed. Some of it goes to feed our animals, like cows and chickens and what have you. Some of that goes to make cornstarch and corn syrup and high fructose corn syrup and all those things you see on labels. 95% of canola, and canola gets used to make canola oil and also like margarine. Okay, that's 95% of the US canola is GMOs. Sugar beets. You might think, huh? What? Well, sugar beets are used to make granulated sugar, aka the white sugar you all have in your pantries. And more than half of the white sugar has been made in this country using GMO sugar beets. Some other things, a lot of the potatoes we buy have been modified, a lot of the summer squash, apples, papayas. What have they done to it? Various things like apples, some of them have been modified so they don't go bad so fast, or they don't brown so fast once you cut them open. Potatoes, so they don't get those like dark spots in them, like they've been modified, okay? So these are just some practical examples of how modified organisms have already been used in our society. We're going to be learning more about it throughout this unit, but hopefully now you have a little bit of a handle on exactly what it is, some of the steps involved, and again, we'll be going through these steps together more in depth in person because you will be doing a lab where you are going to actually genetically modify an organism, okay? So get excited. Hope you took good notes. Make sure you upload them.